The overall goal of this procedure is to determine the ability of ovarian cancer multicellular spheroids to invade a mesothelial monolayer. This is accomplished by first incubating red fluorescent protein-expressing ovarian cancer cells in low-adhesion culture plates to form multicellular spheroids. The next step is to plate green fluorescent protein-expressing mesothelial cells on fibronectin-coated glass-bottom culture dishes to form monolayers. The final step is to transfer the ovarian cancer spheroids from the low-adhesion plates to the plates containing the mesothelial monolayers. Ultimately, fluorescence time-lapse video microscopy is used to quantitate the relative ability of control and experimental ovarian cancer spheroids to form a hole in the mesothelial monolayer.
The main advantage of this technique over existing endpoint assays is that a combination of fluorescently labeled cells and time-lapse video microscopy is used to monitor the dynamics of mesothelial clearance over time.
Prior to the formation of ovarian cancer spheroids, it is necessary to prepare low-adhesion 96-well round-bottom culture dishes. To produce these culture dishes, add 30 microliters of poly-HEMA solution to each well of a 96-well cell culture dish. Incubate the 96-well plates in a 37 degrees Celsius non-humidified incubator for 24 to 48 hours to evaporate the ethanol, leaving a film of poly-HEMA on each well. This poly-HEMA film prevents cells from attaching to the bottom of the well